Hi, good afternoon, everyone. Welcome to our webinar today. It's our webinar in relation to COVID-19 testing. But first of all, before we get into our webinar today, I just wanted to give all of our attendees a quick overview about Randox Laboratories and a little bit of information about what we do. Some of you will probably already know who we are. So just to begin, excuse me, Randox were founded in 1982. We're a Northern Irish company, but we are a global diagnostic manufacturer. We have customers and customer support in over 120 or 145 countries all over the world. Every year, approximately 5% of the world's population are diagnosed using Randox products. Over 100,000 laboratories globally are currently using Randox products. And Randox are also a key partner of the UK's national COVID-19 testing programme. And on the 15th of February of this year, 2021, Randox processed the 10th million sample for COVID-19 as part of the UK government's COVID-19 national testing programme. And we were able to achieve such a significant milestone in the COVID-19 testing programme. This was based upon an £85 million investment since March 2020 in our facilities and our equipment at our headquarters in Northern Ireland. In Thailand, we're probably most famous for our clinical products like our clinical chemistry QC or Accusera, also our clinical chemistry EQA, which is our RICUS program, and also our clinical chemistry line, which is our RX series of instruments. Today, our focus will be on molecular testing. And for molecular testing, Randox have partnered with and have been a partner for a number of years with Qnostics, who are a leading provider of third-party molecular quality controls. And Qnostics within Thailand are available via our partner within Thailand, Q Bioscience. Okay, so what I'm going to do now is I want to pass you over to my colleague, Dr. Dwaine Vance. Dr. Dwayne Vance is a senior molecular product specialist here at Randox. He's also a COVID-19 laboratory manager. Dwayne headed up our COVID-19 testing program and was instrumental to the process and is one of the leading experts on large-scale COVID-19 testing. So I'm just going to pass you across now to Dr. Vance, who's just going to begin today's webinar.
Okay, my name is Dr. Dwayne Vance and I am one of the senior molecular product specialists here at Randox, and for the last 12 months I have been heavily involved in the development, establishment of the Randox COVID-19 super laboratory, which is part of the UK national testing programme. Randox currently is testing approximately 25% of the United Kingdom throughput for COVID-19 testing. The aim of this presentation is to discuss the importance of third-party molecular controls. Third-party molecular controls is something that Randox uses on a daily basis when conducting their COVID-19 tests. This presentation also aims to provide the audience with examples on how to successfully incorporate the use of a variety of different Qnostic controls into laboratories testing for SARS-CoV-2. Okay, there are a number of different ways in which laboratory managers can monitor laboratory performance. The top 6 ways in which labs can monitor performance include internal quality assessment, internal quality control, external quality control, external quality assessment, such as proficiency testing, which includes QCMD, CPT and staff training exercises, and also the implementation of a high quality, robust quality management system. When one incorporates all of these into the laboratory, this has been shown to improve your performance and therefore can result in maintaining accreditation or the acceptance of certification from regulatory bodies. Within the molecular microbiology laboratory setting, standardization and quality is extremely important. Standardization and quality has been well known and well established within the clinical chemistry world, and molecular biology is no different. Standardization and quality aims to support the following: competence in the diagnostic service provided, increased reliability and reproducibility of patient test results, the compliance with appropriate regulatory results and accreditation bodies. Majority of laboratories are working towards common national standards, for example, ISO, CEN, JCI, and LA. They are different national and international regulatory standards dependent upon the country you're in. The most well-known globally would be the ISO standards, which are International Standards Organization, and CAP, the College of American Pathologists. Internationally, there are increasing regulatory demands within laboratories, and because of the global pandemic which we find ourselves in regards to COVID-19, it's super important that laboratory managers that are working in COVID-19 laboratories have laboratories that are accredited under the ISO 15189, ISO 17025 or equivalent. There is pressure to perform high standards. If high standards are not followed, then there may be consequences of failure, and ultimately this may result in the inaccurate reporting of test results. The risk of non-conformance at the next accreditation audit if poor implementation of high quality standards are not maintained. In some countries, there is a potential loss of accreditation and therefore certification will not be provided upon completion of the auditing process. Molecular labs are no different from clinical chemistry labs. The same robust clinical quality control and quality assessment protocols must be followed within molecular labs as in clinical chemistry labs. Over the last number of years, there is an increase in laboratories' reported use of molecular diagnostics. Even more so, this has been extremely evident since the identification of SARS-CoV-2. Many laboratories around the world have been established to test for COVID-19 molecular testing using real-time PCR. One of the most important ISO recommendations is ISO 15189. Clinical relevance and quality control and external quality assessment requirements. 15189/2012 recommends that the use of an independent third-party control, such as Qnostics, should be considered either instead of or in addition to control materials supplied by the reagent or instrument manufacturer. What this means is that independent third-party controls should be used as well as the first-party controls. I will go into a little bit more detail later of the difference between the first-party controls and the third-party controls and what extra advantages the implementation of third-party controls can have with regards to increased quality control within the laboratory setting. This ISO also recommends that the lab should use quality control materials that react to the examining system in a manner as close as possible to patient samples. Okay, so this doesn't necessarily mean that your control materials should be patient samples, but they should be materials that mimic the true effect of a patient sample. For example, Qnostics is provided in a solution which is very similar to a patient sample, and it's transported in dry ice at -20 degrees, which means it has that added extra advantage over other control materials that may arrive in a lyophilized format and then will need reconstituted. And this reconstitution may result in the sample not being exactly truly representative of a patient sample. Quality control materials should be periodically examined with a frequency that is based on the stability of a procedure. So in regards to COVID-19 real-time PCR testing, it is common practice to include a quality control material in every analytical run that is conducted. So for example, worldwide, the most common way for COVID-19 testing is the utilization of a 69 well plate. Within the 69 well plate, there should be at least one positive control which will test for the full process of the molecular experiment, from extraction, amplification, right through to qPCR detection. I will go into a little bit more detail later on how we at Randox conduct our QC processes. Quality control material, for example, Qnostics. The laboratory should choose concentrations of control materials wherever possible, especially at or near clinical decision values. This means that quality control materials should be selected based on their concentration or their titer. And of course, it's important to note that different qPCR real-time assays will have different limits of detection. So it's important that there are quality control material based at the positive limit of detection area. I would like to just take this opportunity to talk a little bit more detail about the Qnostics product range. You will know from the literature that you received that Qnostics has medium Q controls, which are referred to as your daily controls. There are also molecular Q panels and analytical Q panels. The molecular Q panels contain a high, medium, a low, and a negative, and the analytical Q panels contain a very high, a high, a medium, a low, a very low, and a negative sample. So this ensures that the robustness of your assay is looked at in a way that can ensure that the near clinical decision values are adhered to. The lab has to provide the levels of quality control materials run each day, the frequency of performing the quality control. As a general rule of thumb, one control should be run per analytical run, so one Qnostics control per 96 well plate. This is important to prevent any mistakes that may happen with regards to the process of handling samples. Not only are third-party controls very important for the quality management systems within the laboratory setting, but inter-laboratory comparisons, also known as proficiency testing, is also important. I'm not going to talk too much about EQA or proficiency testing today, but I would just like to bring to your attention that Randox also provides a very wide range of EQA programs for molecular diagnostics. The brand in which we have at Randox is called QCMD, which stands for Quality Control Molecular Diagnostics. If you wish to discuss this in more detail, please contact ourselves at Randox or our colleagues at QS Biosciences. There are numerous regulatory requirements that are currently reinforced to ensure that laboratories are using third-party quality controls. Controls should be independent of those produced by the manufacturer or the test analyzer. Again, we kind of discussed this a little bit earlier. First-party controls do not tell you the full story. First-party controls do not look at the test experiment from a non-biased perspective. Third-party controls are required to look at any potential shifts or drifts that may be present within the lot of reagents in which the laboratory is using. Quality control materials should be different from the calibrator materials to ensure that the QC procedure provides an independent assessment. Again, Qnostics material that we provide by Randox are manufactured in an independent manner. All of the material in which we use to develop our material is traceable to international reference standards. So the product is of high quality, robust and independent. The Qnostics material that we provide falls within all of the standard ISO regulations. Laboratories should use more than one level of IQC materials, for example, a normal level plus an abnormal level. Again, these different levels and concentrations and titers can be all part of the Qnostics Molecular Q Panel and the Analytical Q Panel, which contains a wide range of different concentrations of virus within each file. Each panel also comes with a specificity negative to ensure that there's no crossover contamination or abnormalities identified within your molecular workflow. There are multiple different types of quality control practices. Some labs will be using their quality control material supplied with the kit of reagents, which is referred to as your first party controls, or if they're using other types of platforms such as point of care devices, they'll be using their built-in cartridge controls, which is a type of first party control. It should be noted that in-kit controls and built-in controls are not true third-party controls and could be biased and are not independent. Other labs, instead of a commercial QC, will be making and using their own QC material. And what I mean by this is they will be using patient pools which have been derived within the laboratory setting. Patient pool samples tend to be uncharacterized and do not meet ISO standards for manufacturing, nor are patient pools traceable to international standard reference material. One of the added benefits of incorporating Qnostics material within your laboratory setting is that Qnostics material are fully characterized, they're fully quantified using gold standard droplet digital qPCR, and they're traceable to international WHO standard references. True party controls such as Qnostics have been manufactured independently of the instrument manufacturer as per recommendations by ISO or equivalent of your country. This is an important diagram that I would like to discuss. It's the three important strands of quality of measurement within a laboratory setting, and this is very true and very applicable within a COVID-19 environment. Method validation and verification. Validation and verification of the molecular workflow is extremely important because laboratory managers need to ensure that they're measuring what they're setting out to measure. Are they using a qualitative assay to test for COVID-19? Are they using a quantitative assay to measure COVID-19? And are they using quality control material with known quantification levels? Uncertainty of measurement. How well do laboratory managers know what the result is, what they've measured? The traceability of the result. How can that result compare to the results of others that have been reported? and also commutability. Does the QC material that they're utilizing within their laboratory perform sufficiently like a clinical sample? It's important to note that all Qnostics material behave and mimic a true patient sample. The trending of molecular market has changed over the last decade, and again, because of the COVID-19 global pandemic, the molecular market is moving in a way that has been unprecedented. There is a great move from in-house PCR assays to commercial assays, such as the commercial assays that have been developed by Randox laboratories. There's an increased emphasis to develop technologies that are becoming fully automated. Within the Rundox COVID19 laboratories, we have recently installed a full range of automated robotic systems that use air and water displacement for conducting experimentation. There's also new technologies such as the Randox Discovery, which is an open access automated platform which can do 16 samples per batch, extraction, PCR, and detection. PCR tests have changed in a way that it can determine the presence or absence of multiple different pathogens within one PCR reaction. These syndromic tests are also known as multiplex tests, and we at Randox are an example of a commercial company that have developed a high range of syndromic multiplex PCR tests. Due to the increased number of companies that are developing assays, there are competitive pressures. There's an increased interest in standardization of QC material and therefore increased requirements for accreditation and certification. So molecular markets are moving forward at a rate that has been probably would not have happened if COVID-19 did not exist. So it's vitally important that any molecular laboratories in the world, and including Thailand, that are fully subscribed to molecular EQA proficiency testing programs, but are also using third-party molecular controls such as Qnostics to ensure the robustness of their protocols are as accurate and reliable as possible. This image aims to outline the volume, the array, the mixture and variety of different molecular devices that are currently used within the market. One caveat to this is that the increased number of molecular platforms can also complicate the quality assurance process because different qPCR assays behave in slightly different ways. Different qPCR assays look at different genomic targets. Different qPCR assays have different limit of detection, and the PCR mechanism may also be slightly different. This means that it can be sometimes difficult to have a standardized end result. That is why it's important that every laboratory can utilize third-party molecular controls to derive their own, to derive and determine their own units. Near patient testing, also known as point of care testing, is also becoming more and more prevalent within the molecular diagnostics clinical arena. One of the most newest platforms, which I have circled in a green circle, is known as the Bosch Vivalytic. For those of you that have not heard of the Bosch Vivalytic, I invite you to look it up online, or to talk to us at Randox or to our Thailand partners QS Biosciences. The Vivalytic is a well-known, well-established point-of-care platform that is used for the detection of COVID-19 using gold standard real-time PCR technology. One patient test can be detected for coronavirus within 30 minutes, which is one of the quickest PCR tests in the world. This table outlines the key parameters involved to determine the QC robustness of a laboratory. Accuracy, precision or imprecision, linear reportable range, the limit of detection and limit of quantification, which is your upper and lower limits of a molecular assay, your sensitivity, your analytical and clinical sensitivity, and of course your specificity. It's important to ensure that your results are as robust as possible, and any positive results can be detected by ensuring the qPCR assay has got a high level of sensitivity, and to ensure that the ability to return a true negative result when the target is actually negative, that means that the qPCR assay has got a high specificity. As a general rule of thumb, qPCR assays should have a sensitivity of over 98% and a specificity of over 98%. This is a diagram that aims to show you the stages of an assay product life cycle. So whenever companies such as Randox are creating a qPCR assay, it'll always start from the level of development through to manufacture. Regulatory bodies may, for example, CE marking within Europe or FDA within America or other parts of the world, launching of the product, customer evaluation, and then the routine customer use. During the development of an assay, important parameters must be determined, including the sensitivity and specificity, the LOD and LOQ, inclusivity, exclusivity, and linearity. For the purpose of development of an assay, the requirements of a third-party molecular control is essential. Third-party molecular controls will also be required for your verification and validation process and your customer evaluation process whenever you receive your assay into your laboratory. Third-party molecular controls can also be used for training, to evaluate the performance of an assay, to conduct studies, to ensure your molecular process is fit for purpose, and can be used as a daily run control within every analytical run. This is a similar layout to the previous slide, which shows the clinical relevance of incorporating molecular controls within your verification, validation, and implementation stage within your laboratory. It's important to know that Qnostics material can be used as a way to verify your method, to validate your method, and to ensure successful implementation of your qPCR assay into your workflow. Towards the end of this presentation, I'm going to talk a little bit more about the products specifically and provide you more information on how you can retrieve these for use within your laboratory. So why should you use third-party quality controls? This is a very important slide, and it's a slide that I think we should take some time in listening to. Third-party controls should be used to monitor the precision of your patient results from test kits and non-commercial assays. Third-party controls are aimed at preventing errors by consistently monitoring the molecular workflow. To assess variation between lot of reagents, for example, extraction kit or qPCR kit, and this will help to identify any shifts or drifts that may be present within your lot. To provide an independent mechanism to assure that results are reliable and robust. To be able to apply a just-in-time corrective action to eliminate errors that may have been or could be occurred in the future during the molecular process. Third-party molecular controls such as Qnostics are full pathogen whole controls, which can be utilized from the whole process, from pre-extraction to extraction of RNA and purification of nucleic acid, the PCR experimentation process, and the real-time quantitative PCR pathogen detection fluorescence experiment. All Q-Gnostics material mimic patient samples. And what I mean by this is that Q-Gnostic samples are commutable to that of a patient sample, and that is exactly the type of material and quality of material that we're looking for within COVID-19 laboratories. One of the most common questions that customers, laboratory managers, fellow scientists will ask whenever discussing QC requirements will, how often do I use it? When do I use it? How should I use it? What days of the week should I use it? And to be honest, there's no one answer fits all. There's no one fits all answer to this, because the lab has to provide the levels of quality materials run each day. The frequency of the performing the quality control will also be dependent upon the specific laboratory. For example, if you are a high throughput lab or a low throughput lab, the number of staff that you have, the number of shifts you have, the number of extraction devices you have, are you a 5-day lab? Do you work 7 days a week? Is it a 24/7 lab? So all of these different variables will determine exactly how often you should utilize third-party molecular controls. A general rule of thumb for me whenever I'm discussing with people about using third-party molecular controls is it's better to use them sometimes than not use them any times at all. Okay, so it's important to understand and appreciate that it's important to implement the use of third-party controls rather than not using them at all. Positive and negative controls should be tested each time patient samples are tested. This is to ensure the robustness of your assay, your staff, your platforms, your machines, your devices, and the negative controls should be included because that helps us determine if there's any contamination or any issues that may result in a false positive result. And we need to mitigate all potential mistakes as good and as best we can. At least once each day the patient samples are tested, the laboratory samples must include the following: at least 2 control materials of different concentrations for each quantitative procedure, a negative control material and a positive control material for each qualitative procedure. The complexity of molecular assay workflows. As a general molecular layout, the molecular workflow consists of 3 different compartments. Your extraction, where the COVID-19 RNA nucleic acid is extracted from the swab sample, amplification using your PCR reagent, and then your detection, which may be qPCR, gel electrophoresis, or perhaps other detection methods such as the Randox Evidence Investigator chemiluminescence platform. There are also cartridge-based systems such as the Vivalytic point of care system, where all 3 departments occur within a cartridge inside the point of care platform. The variation in molecular assay workflows can result in variation of end result. I'm going to talk now about a little bit more about internal first-party controls. We are all using internal kit controls. However, it is important to note that first-party internal kit controls do not monitor the lot-to-lot variation, because these controls have been specifically manufactured to work alongside a specific lot of reagent by the manufacturer. Not all kit controls go through the whole process of extraction, amplification, and detection, and that is even more prominent because if you can incorporate third-party controls within your molecular workflow, then you can clearly identify any drift shifts or drifts that may be present within your molecular workflow. May that be due to a machine that needs serviced, staffing that may need retrained, or if the lot of reagents are just not conforming to the previous lot that was purchased. Third-party controls monitor the whole process. So you will see here, this diagram shows that this laboratory has conducted 2 different EQA challenges. Challenge 1 in red and challenge 2 in blue. The challenge 1 EQA has resulted with a figure in the middle blue line. However, towards the second EQA challenge, you will see that the standard deviation has increased to 4.8. So this means that there's been a substantial shift in end result due to the lot and lot changes. If third-party molecular controls were not implemented in this laboratory, these shifts and drifts could not have been identified. Therefore, corrective actions could not be implemented, which means that the robustness of your patient's result may be affected and may need some reassessment. So how can Randox help? What services do Randox offer, and how can our distributors within Thailand, QBS Biosciences, really help here? We can provide a whole mixture of different third-party controls that can be used to evaluate your method verification, to look at validation, linearity, limit of detection, limit of quantification by supplying analytical Q panels, and to look at your daily IQC by looking at our Q control material. We've focused today a lot on this COVID-19, the COVID-19 laboratory, but this slide is to show you here that Qnostics also provide a whole host of different other viruses and bacterial targets for third-party controls. CMV, EBV, HSV1 and 2, flu A, flu B, RSV, HIV, hepatitis, chlamydia, gonorrhea, central nervous system pathogens, and pathogens associated with gastrointestinal diseases such as norovirus and E. coli. I would like to invite you to read the Qnostics brochure for more information on all of the products that we provide to you via Randox and QBS Biosciences. Third-party controls measure accuracy and precision of the entire process. First-party controls don't. Third-party controls determine lot-to-lot variation. First-party controls don't. The Qnostics third-party controls can assess genetic diversity by using evaluation panels, which cover all subtypes. Third-party controls can assess the linearity of a molecular platform. First-party controls can't do this. Third-party controls can detect shifts or drifts of reagents. Again, first parties cannot do this. And also, third-party controls can assess the accuracy and the robustness of the entire molecular process. First-party controls, again, do not have this ability. And also, third-party controls can assess your specificity and your sensitivity of the assay which is being used within your laboratory. I'm going to continue now just talking about a little bit more and give you a little bit more of an insight about the Randox COVID-19 testing lab and how we implemented Qnostics controls within our lab as part of our quality management system. So this slide shows an image of a 96 well plate, and within this plate you will see 4 green dots: well A1, well A2, well H11 and well H12. Okay, so I'm just going to go through each of these little green dots because each green dot symbolizes the addition of a QC material. So well A1 we refer to as a negative extraction control. A negative extraction control is implemented within a 96 well plate to ensure that there's no contamination within your process, specifically your extraction process. Your negative extraction control material can be nuclease-free water, TE buffer, or it can be a specificity negative product, such as the specificity product that can be purchased from Qnostics. Not all negative extraction controls need to be a specific negative product. For example, and perhaps every 10% of plates can be a specificity negative product, and the remaining 90% can be nuclease-free water, for example. The positive extraction control located in well A2. This is your medium Q control by Qnostics. And this Qnostics control was located in approximately about 30 to 40% of plates. We were testing, at Randox we test about 100,000 samples per day, which is 1,000 plates. 1,000. Yes, 1,000 plates. So sometimes you have to look at the costing effect here. So about 25 to 40% of our plates, we implemented a positive extraction control, and we made sure that every extraction device was utilized at least 3 to 4 times per shift, and that every qPCR platform was utilized so that every machine was being investigated multiple times per multiple shift across different operators. Within wells 11H and 1112, we implemented PCR amplification controls. The negative PCR control is always supplied with the real-time PCR kit, and the positive PCR control is provided by is a nucleic acid control, and it's important to discriminate the difference between a positive extraction control and a positive PCR control. Your positive PCR control is there to ensure that your processes worked from extraction to detection. However, the PCR amplification control is also there for an additional purpose to ensure that the test is adequately suitable to pick up a positive result, because your positive PCR control should always be at a level that is on the lower end of the limit of detection. So there is assurances that the qPCR assay can detect a sample which may have a low viral load. It's also important to note that PCR controls are only partial genome, whereas positive extraction controls by Qnostics or whole pathogen controls can be used for any molecular workflow and investigates all gene targets, including the S gene, N gene, E gene, and the ORF genomic region. qPCR assays also come with an internal control, which in the form of a human internal control. The primers and probes are contained within the real-time PCR kit to ensure swab integrity, sample equality, and the appropriate extraction process is being conducted. The molecular Q controls, these are also referred to as your daily controls. So these are the controls that we had within well A2. Q controls are positive one-run controls that our concentration is around the medium clinical level. These samples are intended to help laboratories monitor their molecular diagnostic assay on a run-to-run basis within customer-derived limits. Third-party independent extraction controls will ensure that assay drift is detected, monitored, and managed, allowing the laboratory to continue to provide accurate and reliable results. They also help support a laboratory's regulatory requirements under standard ISO 15189. It is important to note that whole pathogen extraction controls, Qnostics, can be used with any molecular assay, regardless of gene target selected, and that is because that the whole viral genome is included within the whole pathogen product. They monitor the entire molecular workflow from extraction to PCR amplification through to detection. Molecular Q panels. Okay, so this is a panel of samples. The previous slide was a molecular Q control, which was one single product, which was at a medium concentration. The molecular Q panel is a panel of 4 different samples. They include a high sample, a medium sample, a low sample, and a specificity negative sample. Molecular Q panels are intended to help laboratories evaluate their assays' analytical measurement parameters and monitor their molecular diagnostic assay procedures. Molecular Q panels can also be used to support laboratory training and in the assessment and development of molecular diagnostic assays from extraction through to amplification and detection. Qnostics also provide analytical Q panels. You will see from this image that they contain a high, a medium and a low sample. These high, medium and low samples are the same as in the previous product, the molecular Q panel. However, it is important to note that the analytical Q panel also includes a very low sample, a low medium sample, a medium high sample, and a very high sample. So there's a more wide range that can be detected. The analytical Q panels have been designed to cover the dynamic range of an assay, allowing the laboratory to assess the linearity, the LOD and LOQ, that's the upper and lower ranges of their assay. Each panel contains samples containing the dynamic range of most of the molecular assays within the market in a linear progression, all of which should be treated as a patient sample within an assay run. Molecular controls, I'm going now to list a few specific uses to improve quality when using molecular controls. Lot-to-lot assessment of nucleic acid extraction reagents or real-time PCR kit. Molecular controls such as Qnostics should be used to ensure the previous lot of reagents are comparable to the new lot of reagents. Molecular controls are useful in the clinical validation of equipment such as extraction platforms and real-time PCR machines. It is important that validation studies and post-maintenance calibration checks are initiated after installation and after routine maintenance or repair. The training of staff. For example, during the training of extraction scientists, each member individually pipettes 70 water samples and 20 Qnostic Q control samples. This helps determine the pipetting accuracy of the trainee scientist. If all water samples are called undetermined or negative, and all Qnostic control samples are called positive, then the trainee is therefore deemed competent to conduct the extraction process for COVID-19 testing. Over the last 12 months, there has been numerous COVID-19 laboratories set up. There's been many more scientists employed to specifically work within a COVID-19 environment. It is therefore important that these staff are trained appropriately. And one way that we can ensure staff are trained appropriately is the utilisation of Qnostics Q controls as a positive material. Analytical or linearity control panels, such as those supplied by Qnostics, can be used to assess linearity, LOD and LOQ, to cover a dynamic range. When introducing a new or amended workflow, for example, a new extraction platform or a new COVID PCR assay, analytical panels are essential to determine how parameters such as LOD and sensitivity can be positively or negatively affected. This ensures that the best quality and performance can be utilised within your lab, resulting in improved accuracy. So if you're looking at new methods to extract or looking at new methods to determine the detection of an assay, then you will need to conduct an analytical or linearity control panel, such as that supplied by Qnostics. Also, as the number of samples that are processed increases, a lot of laboratories are turning towards the use of automated liquid handling robot systems. Therefore, it is also important to test that robotic systems produce equal or better results than manual processes. And in Randox, we utilised the Molecular Q Panel, which is low, medium and high samples, to ensure that there was no crossover contamination whilst using the robots. So we knew that low samples had a specific CT value, high samples had a specific CT value, medium samples had a specific CT value, and negative samples, of course, had a 0 CT value. So therefore, it is important to use quantifiable, traceable material to ensure your liquid handling robotic systems are working at an adequate level. Quality control material can also be used for R&D processes. For example, within Randox, we use QNostics as part of our research and development when we developed our own total nucleic acid extraction kit. And when we developed our own qPCR assay, we also used QNostics as our control material. This gave us confidence that our assay was working to the highest possible standard, and our sensitivity and specificity results were as accurate and robust and as best as possible. This slide is a few screenshots of the SARS-CoV-2 products from QNostics. On the top of the screen, you will see the SARS-CoV-2 medium Q control, which is your daily control. Your medium Q control is a whole pathogen control designed to monitor assay performance on a run-to-run basis. As true third-party controls, assay drift is detected, monitored and managed. This control is stable for up to 5 days. Okay, so that's one of the benefits of this product. It's got 5 days stability. It comes with 5 vials, and each vial's got half of a mil. So this product can be utilized over a course of 5 days within a daily setting. The Molecular Q Panel. It's also important to note that the medium Q control is CE marked. The SARS-CoV-2 Molecular Q Panel provides a low, medium and high sample and is also CE marked. The Analytical Q Panel is designed to span the analytical measuring range of your assay to determine linearity, LOD and LOQ. And there is a total of up to 9 different samples, ranging from very, very low to very, very high. If you would like more information on how to order these products, please contact us at Randox or consult with our partners, QBS Biosciences. We also have a multiplex respiratory control. This control is a multiplex control that can be utilized as part of any syndromic testing or multiplex PCR assays that are currently out in the market, such as your Cephagene Expert, your BioFire, your Vivalytic, your Roche Liat. This control includes 5 vials, and within each of the 5 vials contains the presence of 4 different pathogens: SARS-CoV-2, influenza A, which is your H1N1 subtype, influenza B Victoria lineage, and your RSV. This is a popular product on our QNostics multiplex respiratory controls.